Hi, everyone. Today my presentation will be called Through an Intern's Eyes, an Intern's Learning Experience. Sorry. No worries. Hello, everyone. I'm Sandy, and I'm a recent graduate from NYU, where I studied biology and computer science. I have a strong interest in bioinformatics and computational biology. This summer, I was very, very fortunate to be able to intern at the Columbia Center for Precision Medicine and Genomics. And some of my main responsibilities is to use bioinformatic tools to understand kidney disease, to analyze and interpret genomic data in the context of biological concepts, to contribute to the research and the development of novel bioinformatic approaches. And one thing that I noticed is a really big and unexpected part of my internship is actually to work as a team alongside other interns and aid in the support of their progression. So as we all know, humans are usually born with 2 kidneys. And kidneys are important because they help remove waste and toxins from the body. They also help to balance our bodily fluids, and they can play a role in regulating blood pressure and the production of red blood cells. However, some people can be born with 1 or 2 abnormally small or malformed kidneys. This image here will show you what a small and malformed kidney looks like next to a normal sized one. So my project uses collapsing analysis to identify the potential disease-associated genes responsible for this type of kidney disease. How it works is that we use the convenience of bioinformatic tools and the availability of large-scale sequencing data in order to count how many variants that are deleterious that are in the cases, which are the individuals who have the disease versus the control, or the individuals who do not have the disease. And by quantifying the number of deleterious variants between cases and controls in the same genes for every single gene, we can see which genes are likely to be the cause behind this kidney disease. So in this example right here, you can see that in the cases, we have 3 deleterious variants in this gene. However, in the controls, we only have 1 deleterious variant in this gene. Therefore, we can say that there's a high probability that this gene is responsible or associated with this disease. And so my project is using this type of analysis to find the disease genes behind the specific kidney disease. My hypothesis is that we will be able to use collapsing analysis with a public control data set specifically, such as NOMAD, to successfully determine disease-associated genes behind kidney diseases. At my internship, I learned a lot of bioinformatics skills, but I also learned a lot of skills for internships and jobs in general. So I learned that research projects take a lot of time. It can take 6 to 8 weeks to get any meaningful data. I also learned how to conduct literature review and use that information as my primary resource for my ongoing research project. I also learned how to use new programs that I never used before, such as VDP Docker for genome annotations. And I also got more accustomed with working with larger data sets and also with using the command line. I also learned how to present scientific data and also how to work as a team. But I think my summer internship can be summed up with this quote by John Shedd, and that is that a ship is safe in harbor, but that's not what ships are for. In other words, sometimes during your internships, you may be met with challenges, but that is okay. Learning how to overcome these challenges is actually part of your working experience and learning experience. So I would like to conclude this presentation by taking the time to thank our LifeSci people such as Sally, Rich, Anita, and Arlie. Thank you so much for organizing today and all our events this summer. Thank you for helping us get jobs. And thank you to Dr. Sheila Razuli Sarab and Dr. Nick Sears for being my mentor, Dr. Garabi for accepting me as an intern, and thank you for Columbia University Irving Medical Center for letting me work there this summer. I'll be happy to take any questions if you have any.
Today's presentation, can you describe a challenge that you faced and how you overcame it?
Yes. So actually, one of the challenges I faced was learning how to annotate my data. So when you begin with a new project, sometimes your computer is not equipped with the same technology that you might need for your project. One of the things that I needed was VEP, which I need to annotate my genome and data. But I was trying really hard, and for some reason, I was having a lot of difficulty installing it. And so my challenge would just be finding errors and finding these roadblocks and trying to figure out ways to overcome them. Eventually, I landed on using VEP Docker, which is a container for VEP, which has everything installed for VEP. And that made it much easier to download VEP. However, it has different functionalities and ways that you work with it. So yeah, that would be my main setback, because technical issues can delay your results a lot, but learning how to overcome them and finding different ways to resolve your technical issues is part of the learning process, especially if we're doing something in tech. Thank you for listening.